Ever notice how in cancer treatment, doctors can now pick drugs based on a tumor's specific genetic signature? That's precision medicine. But what if I told you this same revolution is happening right now, far beyond oncology, and it's transforming how we treat everything from autoimmune diseases to Alzheimer's? And the secret ingredient isn't just new drugs, it's something far more fundamental: the tests we use to understand disease. Let's break this down. For years, the biggest hurdle in developing precise treatments for non-cancer diseases was a lack of ideas or potential drug targets. It was a lack of good tests, what we call assays. We couldn't accurately measure the complex biology inside a patient, connect that biology to who would get sick, or see if a drug was actually working on a molecular level. We were flying blind, relying on symptoms alone. But that's all changing. Think about it like upgrading your phone's camera. You go from a blurry, single-color image to a high-definition, multispectral video. That's what's happening with medical assays. As these technologies mature, they're not just improving diagnostics, they are fundamentally rewriting the playbook for drug development. Let's dive into some of the most exciting areas. Take immunology and inflammatory diseases like rheumatoid arthritis or Crohn's. We used to lump patients together based on their clinical symptoms, like joint pain or gut inflammation. Now, we're shifting from that broad phenotype to a specific molecular endotype. We can pinpoint exactly which biological pathway is driving the disease in an individual. How? The assays have evolved dramatically. We've gone from measuring a few clunky proteins in the blood to running multiplex panels that measure hundreds at once. We've moved beyond basic cell counting with flow cytometry to technologies like CITF and single-cell RNA sequencing, which give us an incredibly detailed snapshot of every single immune cell and what it's doing. We can even take a patient's cells, stimulate them in a lab dish, and see exactly which inflammatory pathways light up. This has a massive impact on drug development. Instead of giving everyone the same drug, we can stratify patients by their dominant pathway. Is your inflammation driven by IL-17, or is it an IF-gamma problem? We can now design clinical trials that enroll only patients whose biology matches the drug's mechanism. This allows us to find the right dose faster, see a clear signal of effect, and even rescue drugs that previously failed in broad populations because they only worked for a specific molecular subgroup. AUP. Now let's turn to the brain. Neurology and psychiatry have long been the final frontier of medicine, defined almost entirely by symptoms and observation. Diagnosing conditions like Alzheimer's or depression was based on checklists, not biology. But here too, new assays are cracking the code. We used to rely on invasive spinal taps to measure a few proteins in cerebrospinal fluid. Now, with ultra-sensitive technologies like Simoa, we can detect those same markers of neurodegeneration in a simple blood test. At incredibly low concentrations, we have advanced PET scans that can light up specific pathological proteins like amyloid and tau directly in the living brain. We are combining genomic risk scores with brain activity patterns to identify subtypes of disease that look the same on the surface but are biologically very different. For drug developers, this is a game changer. We no longer have to wait years for a patient's memory to decline to see if a drug is working. We can use these fluid and imaging biomarkers to get an early readout on target engagement. Did the drug hit its mark in the brain? Is it lowering the bad proteins? This enables much faster go or no-go decisions, saving billions of dollars and years of time. It allows us to finally dissect heterogeneous diseases like Alzheimer's and multiple sclerosis into biologically meaningful subpopulations, paving the way for targeted treatments. This pattern repeats across medicine. In rare and genetic diseases, we've moved from diagnosing based on a collection of strange symptoms to pinpointing the exact molecular defect. We went from single gene tests to whole genome sequencing, which can scan a person's entire DNA blueprint. This means a molecular diagnosis is now the entry ticket to a clinical trial. Trials can be incredibly small but powerful, focused on patients with a specific mutation. And approvals can be accelerated because we can directly measure if a gene therapy is working at the molecular level. Even in common cardiometabolic diseases like heart disease and diabetes, the shift is on. We're moving beyond simple risk factors like cholesterol and blood sugar. We're using proteomics to measure hundreds of proteins in the blood, metabolomics to map out metabolic pathways, and genomic scores to understand inherited risk. This helps us identify who will respond best to a certain drug, who might have side effects, and allows us to target specific mechanistic pathways rather than just treating the symptoms. So what are the big technology trends driving this sea change? First, assays are evolving. We've gone from single measurement tests to multiplex and multi-omic platforms that capture a whole system at once. They've become quantitative and can be tracked over time. Second, our sampling methods are getting smarter and less invasive. The era of painful tissue biopsies is giving way to the liquid biopsy analyzing blood, urine, or saliva. This allows for serial monitoring, so we can track a patient's disease dynamics over time, not just get a single, static snapshot. And third, our ability to analyze this mountain of data is exploding. We're moving from manual interpretation to using machine learning to find complex patterns that no human could ever see. We're replacing simple positive or negative cutoffs with sophisticated models that calculate an individual's probability of risk or response. Biomarkers are no longer just descriptive data points. They are powerful decision-making tools. This completely reshapes the drug development strategy. The traditional model was to enroll a broad group of patients, hope for the best, and wait for a late-stage signal of efficacy, leading to a massive failure rate in phase 2. The new precision model uses biomarkers to enrich for the right patients from the start. It gets an early signal of whether the drug is hitting its target, allowing for faster decisions to kill a failing project or accelerate a winner. It moves from one-size-fits-all dosing to dosing informed by an individual's biology from a regulatory perspective. Agencies like the FDA are embracing this. Biomarkers are increasingly being used to enrich trials, to show a drug is working pharmacodynamically, and in some cases, even as surrogate endpoints for approval. The focus is on ensuring the test is analytically valid and that we understand its clinical context and interpretation. So, what's the key takeaway? Precision medicine beyond cancer is finally being unlocked. And it's not just about discovering new drugs. It's being driven by the incredible maturation of assay technologies. These tests allow us to see disease with a clarity we've never had before, to define its biology, stratify patients, and measure response in a way that is truly clinically actionable. As our tools to see get better, our ability to treat gets smarter. This is the future of medicine, and it's happening right now. Thanks so much for watching. If you found this breakdown helpful, be sure to like this video and subscribe for more deep dives into the future of health and technology. See you in the next one.